Ladies and gentlemen, it's really great to welcome all of you here. It's been a few years since it was like this. So very welcome to Nobel Forum and for the announcement of this year's Nobel Prize in Physiology or Medicine. My name is Thomas Perlman. I'm the Secretary of the Nobel Assembly. I will first read the announcement in Swedish, followed by English, and we will then present the background to the prize and then open up for questions. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2022 skall tilldelas Svante Pääbo för hans upptäckter rörande utdöda homininers arvsmassa om människans evolution. The Nobel Assembly at Karolinska Institutet has today decided to award the 2022 Nobel Prize in Physiology or Medicine to Svante Pääbo for his discoveries concerning the genomes of extinct hominins and human evolution. Here he is. The laureate, Svante Pääbo, was born here in Stockholm, in Sweden, in 1955. He performed his prize-winning studies at the University of Munich in Germany and at the Max Planck Institute for Evolutionary Anthropology in Leipzig in Germany. So now I turn to Anna Wedell, a member of the Nobel Committee, who will now describe the discovery. So please, Anna.
Thank you. My glasses. This year's Nobel Prize concerns human evolution. Humanity has always been intrigued by its origins. Where do we come from, and how are we related to those who came before us? What makes us different from hominins that went extinct? Before Pääbo's discoveries, knowledge was derived mainly from paleontological and archaeological findings. Bones and artifacts from extinct relatives were found at multiple sites around the world. One famous example is the Neanderthal, discovered in 1856. Many questions, however, could not be answered by analysis of ancient bones and artifacts alone. Questions such as how we and our extinct relatives are related to each other, and, importantly, what makes us different. DNA technology offered a new way of addressing these intriguing questions. DNA holds the code of life. Gradual changes to this code, mutations, form the basis of evolution, and these mutations remain in our DNA as traces from our ancestors. Our cells contain DNA in two different compartments. The long nuclear DNA carries the overwhelming majority of our genes, but the small mitochondrial DNA is present in thousands of more copies and therefore easier to study. Svante Pääbo was from an early age fascinated by ancient human history. While using DNA technology for his doctoral studies in immunology, he also attempted, as a side project, to use these modern tools for studies of ancient human remains. He soon realized that this is extremely challenging since ancient DNA is damaged by chemical modifications. It is also fragmented and present in extremely low amounts together with contamination from microorganisms and present-day humans. Svante Pääbo therefore embarked on a long series of rigorous technological developments that lasted over 3 decades. He performed much of this work on extinct animal species, but his goal was a seemingly impossible task. He aimed for our closest relative, the Neanderthal, that went extinct around 30,000 years ago. After setting new experimental standards in this challenging area, Pääbo first set out to retrieve and analyze Neanderthal mitochondrial DNA. He obtained a bone from the Neanderthal type specimen that had been discovered in 1856. From a small piece of this bone, he managed to extract and sequence mitochondrial DNA. He compared his new sequence to present-day humans and to chimpanzees and found that the new sequence was different from both of them. This was an important milestone. Pääbo had proved that it is possible to retrieve and sequence DNA from 40,000-year-old bones. Mitochondrial DNA, however, contains very limited information, and Pääbo's ultimate goal was to sequence the 3 billion base pair Neanderthal nuclear genome, which would allow in-depth analysis and comparisons across multiple genomic regions. The human reference genome had been sequenced in a huge international collaborative effort, and the chimpanzee reference genome sequence was also available. Sequencing the nuclear genome of an extinct hominin, however, seemed impossible. Pääbo embarked on his bold plan, and for this he needed more bone. Thanks to his strong international networks, he managed to obtain 3 bone specimens from Vindija cave in Croatia, together with additional material from Germany, Russia, and Spain. All along, he refined and optimized his experimental procedures. By skillful use of new high-throughput sequencing technologies and by bringing together a large international group of collaborators for advanced computational analysis, Pääbo finally achieved the impossible, sequencing and assembly of the Neanderthal genome. For the first time, important questions on our relationship with the Neanderthal could be addressed. The time of divergence could now be determined to around 800,000 years ago. Pääbo and his team also surprisingly found that gene flow had occurred from Neanderthals to Homo sapiens, demonstrating that they had children together during periods of coexistence. Importantly, comparisons could now also be made to search for differences between Neanderthals and Homo sapiens, differences that may underlie features that are unique to all humans living today. Pääbo's team also retrieved DNA from a minute finger bone found in Denisova cave in the Altai Mountains in southern Siberia. Again, they started by analyzing mitochondrial DNA, and to their surprise, the sequence was different from both present-day humans and from Neanderthals. Pääbo had discovered a completely new hominin entirely by analyzing and comparing genome sequences. This was a sensational discovery. The new hominin was given the name Denisova. Pääbo's team next analyzed the nuclear Denisova genome sequence, enabling important investigations. Neanderthals and Denisovans were shown to be sister groups, diverging from each other around 600,000 years ago. They further found evidence of gene flow from Denisova into Melanesian Homo sapiens, indicating that mixing had occurred. Svante Pääbo's groundbreaking discoveries have provided important new knowledge regarding our evolutionary history. Neanderthals and Denisovans lived in different parts of Europe and Asia for hundreds of thousands of years and adapted to these environments, while Homo sapiens evolved and lived in Africa. By mixing with them after migrating out of Africa, Homo sapiens picked up sequences that improved their chances to survive in their new environments. We now know that present-day humans in Europe and Asia can carry 1 to 2% of Neanderthal sequences, and that Asian and Southeast Asian populations carry an additional up to 6% Denisova sequences. Examples include a gene variant that improves living at high altitude in Tibetans, and sequences that affect how our immune system responds to different infections. No extinct hominin DNA has yet been sequenced from Africa, since conditions for preserving archaic DNA are poor in tropical climates. Thanks to new advanced computational methods, it has still been possible to find indications of gene flow from as yet unknown archaic hominins into Homo sapiens on the African continent. Through his persistent and innovative work, this year's Nobel laureate established a new scientific discipline, paleogenomics. This area is now expanding dramatically. After the discoveries cited here, Pääbo went on to generate genome sequences from many additional Neanderthal and Denisova individuals. In parallel, genome sequences from ancient Homo sapiens have also accumulated from different time points and different geographic locations by research groups around the world. By using Svante Pääbo's archaic genome sequences for comparative studies, important lessons are being learned regarding our evolutionary history, including how we spread across our planet. Svante Pääbo's groundbreaking discoveries allow us to address one of the most fundamental questions of all: what makes us unique? Like us, Neanderthals had big brains, they lived in groups, and they used tools, but these changed very little during hundreds of thousands of years until the Neanderthals, for unclear reasons, disappeared. Homo sapiens, on the other hand, rapidly developed complex cultures, figurative art, and advanced innovations. They crossed open waters and they spread to all parts of our planet. The basis for this dramatic development must lie in genetic changes that occurred after we separated from Neanderthals and Denisovans not so long ago. By providing genome sequences from our closest extinct relatives, Svante Pääbo has provided a novel, much more recent reference point for studies of who we are and where we came from. Thanks to his seminal discoveries, genetic variants that are shared among all now living humans, but absent in Neanderthals and Denisovans, have been identified. The list of such genetic variants is not very long. Understanding their functional implications is a focus of intense research by Svante Pääbo himself, but also by other scientists. Thank you.